Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Nabi bin Ahmad Basri. Today I will share about my final year project, development of surface acoustic wave device for controlling cell migration. Before that, I want to thank my supervisor, Mr. Mazli bin Mazalan, for guiding me in this project. This video will divide into three parts. First part is introduction, second part is literature review, and the third part is methodology. For the introduction part, I will talk about the project background, problem statement, and objective of this project. In living organisms, collective cell migration plays key role in various physiological and pathological processes. During wound healing, collective of cells will migrate and cover wound site. On the other hand, the dysregulated cell migration may contribute to development of congestive heart failure during cardiac fibrosis. The application of an acoustic wave can accelerate cell migration by regulating the expression of migration-related proteins. Before the acoustic wave technique have been found, numerous research have demonstrated that chemical, mechanical, and electrical stimulation give effect on the collective cell migration behavior. The control of cell migration using chemical, electrical, and mechanical has several limitations that prevent its potential therapeutic method, and it also not suitable to implemented in medical facilities. The purpose of this project is to overcome the limitation that have been mentioned before. So the objective for this project is first, to fabricate SAW device for controlling cell migration. Second, to characterize and testing SAW device. And third, to analyze the velocity of cell migration on SAW device. Next is literature review. In this part, I will talk about the first, cell migration. Second, the mechanical factor for cell migration. Third, surface acoustic wave device. And lastly, I will talk about the type of IDT used in SAW for live cell studies. Cell migration. Cell migration is regulated by biochemical gradient, electrical field, or geometric curvature. Previous studies revealed that mechanical stimulation can regulate cell migration by modulating the force balance in a cell. A driving force for cell migration is primarily generated by ectomycin system in the cytoskeleton. Mechanical factor for cell migration. Most of previous study on cell migration use chemical variable. At the same time, it was reported that mechanical stimulation can be control cell migration, such as electrical cue, substrate stiffness, cyclic tension, and fluid stress. However, these stimulations are based on invasive procedure. Surface acoustic wave device. The acoustic wave technique was firstly documented by White and Bodmer with the introduction of IDT in 1995. They show that the IDT, which consists of specially periodic thin film metal electrode over quartz substrate, that can generate a frequency traveling across the surface acoustic wave. Previous research by Imashiro, he found that propagating acoustic wave can enhance or suppress the speed of cell migration depending on the wave intensity. Imashiro used voltage at the range 0 volt to 18 volt, but for my project, I will use a different voltage that is more than 18 volt. There is several advantages of SAW device to implement it in the medical facilities, such as first, easy application for tissue in vivo. Second, biocompatibility. Third, non-invasiveness. Second, fourth, directionality, and lastly, fast delivery. Type of ADT used in SAW. There is 6 types of ADT used in SAW technique for life cell study. First is conventional IDT. Conventional IDT is the first and the most simple IDT that is composed of straight rectangular finger which lie on piezoelectric substrate surface and connected to pad at each end. This structure creates a range of electrical field that alternate direction from transducer finger pair and in turn generate the inverse piezoelectric effect to alternate areas of compressive and tensile stress within the substrate. Second is single phase unidirectional transducer SPUDT. It based on double electrode IDT. This type of IDT is frequently used when the frequency response need to be controlled precisely. Third is slanted fingers IDT. The slanted finger IDT widely used in the biomedical field, especially to handle the droplet and particle. Number 4 is focus IDT. Focus IDT were capable of focusing the SAW, whereas the focus IDT with the wave surface shape exhibited better focusing properties. Number 5 is chirped IDT. The chirped IDT has a linear gradient in the finger distance that allows the SAW to resonate on a wide range of frequencies by tuning the input signal. And lastly is multiple IDT. Multiple IDT allows multiple properties of a thin film material to be simultaneously extracted to achieve more complete characterization than when using a simple SAW device. In principle, this would enable the fabrication of multiple SAW systems capable of operating in both gas and liquid phase. Next, for the last part is methodology. In this part, I will talk about the lift-off technique, electron beam evaporation, surface profiler, network analyzer, and cell observation on inverted microscope. For my project, I will use the lift-off technique to fabricate the SAW device. A step need for the fabrication of the SAW device is first coating the negative resist. Second, ultraviolet exposure on negative resist via photomask. Third, develop negative resist. 4. Deposit metal layer. And lastly, lift-off process by Akiton. For my project, I will use electron beam evaporator to deposit aluminum layer on substrate as IDT. The thickness of metal layer that can be deposited by the EB machine is 100 nanometer. And for my project, the thickness of aluminum required is only 400 nanometer. Before I deposit the aluminum layer on my SAW device, I will test on 4 dummy substrate. Surface profiler was used to measure the thickness of aluminum layer after it finished deposit by the E-beam. When the thickness of aluminum layer is 400 nanometer, the SAW device will be completed by lift-off process. After the SAW device is complete, it will be tested by applying voltage. Network analyzer is used to test the properties of SAW device. First is resonance frequency, second is the amplitude wave. After confirmed SAW device can operate well, a cell will be put on the center of the SAW device. Inverted microscope will be used to observe the cell migration. Different input voltage will be used to analyze the velocity of cell migration. That's all from me. Thank you.